The broadcast is now starting. All attendees are in listen-only mode.
Hello, and welcome to Breast Cancer Action's webinar, Breast Cancer and the Environment, the Growing Evidence. Thank you so much for joining us today. My name is Saru Kaiser, and I'm the Education and Mobilization Coordinator here at Breast Cancer Action. Joining me on the webinar today will be Rudel, Director of Research at Silent Spring Institute, and Dr. Megan Schwarzman, Research Scientist at the Center for Occupational and Environmental Health at the University of California, Berkeley. A few quick announcements before we get started. Breast Cancer Action doesn't take money from any corporation that profits from or contributes to the breast cancer epidemic. Our work is largely supported by individual donors. Please consider making a $25 donation today to support work like these educational webinars. The presentation of Breast Cancer and the Environment, the Growing Evidence, will last about 45 minutes. At any time during the webinar, you can ask a question by typing it into the question bar. We'll save time at the end to answer questions. And remember, you can type this in any time while we're going through. We want you to get involved with Breast Cancer Action. This webinar is a great way to do that. Please stay tuned for other ways we'll mention later on. I want to go through our agenda quickly. Today we're going to talk about what's being done to reduce exposures to toxic chemicals. We're also going to talk about how we're studying environmental links to breast cancer, about how improved methods for identifying toxic chemicals are being developed, and also a little bit about what you can do to strengthen chemical safety in the U.S. Now, Breast Cancer Action was founded in 1990 by a group of women who were frustrated with the lack of information about breast cancer. Our founders, only one of whom is still alive today, knew that their private medical crises were part of a larger public health emergency, and that the experiences of those dealing with breast cancer really needed to be heard to address the crises. Breast Cancer Action's mission is to carry the voices of people affected by breast cancer to inspire and compel the changes necessary to end the epidemic. Our advocacy is conducted through a social justice lens because the politics and policies of breast cancer disproportionately affect poor women and women of color. Our independence from pharmaceutical company funding puts us in a unique position to address issues of health equity and exposures to toxins in our environment, as well as to put the needs of patients before pharmaceutical company profits. We have three main program priorities. The first is putting patients first, where we advocate at the Food and Drug Administration in favor of treatments that are less toxic, more effective, and less expensive than those already available. We also provide information about breast cancer to anyone who needs it. The second is creating healthy environments, where we work to reduce the involuntary exposures people encounter that put them at risk for breast cancer by holding corporations accountable for unhealthy products and practices. We also support legislation that would better protect us from chemicals in our environment and would make personal care products safer. And the last priority is eliminating social inequities related to breast cancer, where we work to create awareness that it is not just genes, but a complex interplay of economics, power, racism, and discrimination that lead to disparities in breast cancer incidence and outcomes. Our first presenter, Rudel, is the Research Director at Silent Spring Institute, where she leads major exposure and toxicology research programs focusing on endocrine-disrupting chemical exposures and on mechanisms by which chemicals may influence breast cancer risk. Silent Spring Institute is an independent, nonprofit scientific research organization founded in 1995 by breast cancer advocates wanting to create a lab of their own to do research on environmental factors and breast cancer to identify opportunities for prevention. Silent Spring is funded by government and foundation grants and charitable donations and often conducts its work in collaboration with academic scientists. Silent Spring Institute publishes their work in the peer-reviewed scientific literature and also makes it accessible to advocates and the public. Welcome, Ruth Ann.
Thank you.
Our other presenter, Dr. Megan Schwarzman, focuses her work on endocrine-disrupting substances, reproductive environmental health, U.S. and European chemicals policy, and the implications for human health and the environment of the production, use, and disposal of chemicals and products. She is a research scientist at the Center for Occupational and Environmental Health at UC Berkeley School of Public Health. and the Associate Director of Health and Environment for the Interdisciplinary Berkeley Center for Green Chemistry. She earned her medical degree from the University of Massachusetts, completing her specialty training in family medicine at the University of California, San Francisco. And she earned a Master's of Public Health from the University of California, Berkeley. Dr. Schwarzman also practices medicine part-time at San Francisco General Hospital. Welcome, Megan.
Thanks. Glad to be here.
And now I'm going to turn it over to Rudel.
Hello, everybody who I can't see, but I trust you're out there. I am very happy to be here to talk a little bit about what we've been doing at Silent Spring Institute to learn about how environmental chemicals might affect breast cancer and to identify opportunities for prevention. Silent Spring Institute was founded almost 20 years ago now by the Massachusetts Breast Cancer Coalition as essentially a lab of their own, our own research organization to do research on this question in particular, which they had felt wasn't being adequately addressed by sort of the larger cancer funders. So over the last few years, I'd say maybe the last 5 years, there's been a renewed attention to what are the opportunities to better predict what chemicals might cause cancer and to prevent it. This report came out from the President's Cancer Panel in 2009, and they acknowledged, I think, two things. One, that the true burden of environmentally induced cancer has been underestimated, and also that we need to find methods to predict what chemicals are of a concern for cancer, that human evidence is often too difficult to get, that we can't wait for human evidence. And I'm going to talk a lot about that concept in this presentation. Some background on breast cancer. So looking across the world, breast cancer is the most common cancer in women. The incidence rates are higher in developed compared to developing nations and rising rapidly in developing nations. And risk changes when women migrate. So women who move from a area of lower breast cancer incidence to an area of higher breast cancer incidence, their breast cancer risk sort of increases until it reaches in their daughters perhaps or their grandchildren the rate in the place that they moved to. So these observations suggest that whatever the factors are that make breast cancer rates go up, maybe some of them we can intervene on to try to prevent some of the breast cancers. Looking at what we know about risk factors for breast cancer can give us some clues about where to look for other preventable causes. Besides family history, most of the rest of the risk factors are related to are due to exposure to something that you would call a sort of a classical carcinogen, like a ionizing radiation, something that damages DNA, or factors that are related to hormones, including reproductive history, body weight, use of pharmaceutical hormones and alcohol, lack of physical exercise, and tobacco smoke. And that so those observations give us some ideas about where to look when we're thinking about environmental chemicals. Everybody wants human evidence in order to take action, because sometimes the action might be characterized as economically expensive. But that evidence can be very hard to get. And so thinking about, well, what kinds of studies tell us that something causes cancer? What you need to have is a large group of people followed for a long period of time, where you have high, well-defined exposures for each person in the group. You you know they had the exposure, they didn't have the exposure, and then 15 or 20 years later, you know they had the disease or they didn't have the disease. And you sort them into these 4 boxes: disease, no disease, exposed, not exposed, and count. And then you determine whether there's a higher risk of disease among the exposed compared to the unexposed. But it's hard to get that kind of data where you have followed and you have good information about exposures. Most known human carcinogens that are from chemical exposures, those have been identified in occupational studies, and they've primarily been studies of men, and as a result, they've primarily been for cancers other than the breast. But there are quite a few chemicals that have been identified through history as carcinogens in occupational studies. And one of the reasons that this has been primarily through the study of men, is that traditionally men have had a different kind of work history than women. They might more often have been in one job for a long period of time, or one career, and also more often would be having a pension and health insurance. And those two sort of administrative activities lead to record keeping, and that kind of record keeping leads to the ability to go back and make a chart, like the one on the previous slide, where you can sort people about their exposure and whether they got the disease or not. But if you don't have any kind of record keeping like that, it's difficult to do. Now there is an occupation in a women's occupation that was recognized in the 1700s as being associated with higher breast cancer rates, and that is nuns. And that observation led to the really a lot of focus on reproductive risk factors being important for breast cancer risk. These nuns didn't have kids generally, and compared to other women of that time who had them often and early. The other kinds of studies that have been the basis of identifying cancer causes would be accidents or disasters like atomic bomb and for ionizing radiation, and the study of pharmaceuticals like hormone replacement therapy or medical radiation. Both of those have shown associations with breast cancer. So those are the kinds of studies that we that tend to reveal cancer causes. Now, DES, diethylstilbestrol, is a synthetic estrogen that was used prescribed to pregnant women in the 50s and 60s, ostensibly to prevent miscarriage, which it didn't do, but it did lead to a variety of other adverse health effects in the children of the women who were exposed during pregnancy. The first sort of observation that was made is of a very rare vaginal cancer in teenage girls, the daughters of the exposed women. And then there have been some studies looking at the increased risk of breast cancer. But the first lawsuit for association between DES and breast cancer was just settled in 2012, very recently. And thinking about why was that? Well, it took over 60 years to develop human evidence of a link between DES exposure in utero and breast cancer, as it took 60 years for those cancers to develop. And just in 2011, 60 years after these women were exposed in utero, was a paper published that I'd say was pretty conclusive that it increased risk of breast cancer, and that was what was the basis for the lawsuits. But the point I'm trying to make here is just that this isn't a practical way to figure out whether chemicals in the environment cause breast cancer. We don't really have 60 years to wait. We can't actually track exposures very well, and we're trying to make decisions about wise chemical use now. There's a quote from the International Agency for Research on Cancer that every group 1, that's a known human carcinogen, every group 1 agent could be considered to represent cancers that might have been prevented had scientists been able to predict cancer hazards earlier, or had public health authorities been willing to act more quickly when scientific information became available. So often we have evidence from laboratory studies, animal studies, that suggest the chemical might cause cancer. In some cases, we've waited to act until we had evidence in humans, and the result of that has been a lot of cancers that could have been prevented. So we at Silent Spring, we operate within this framework for cancer prevention science, which is, okay, how are we going to predict which chemicals we should be concerned about? And we look for a biological mechanism of how it might cause cancer, might cause breast cancer, and human exposure. And when we have information that suggests we've got both of those things going on, and that's a basis for action, which could be educating manufacturers or consumers. It could be regulation. It could be asking manufacturers to reformulate. And probably there are other kinds of action as well that are possible. There are 3 biological mechanisms that we have identified and targeted. The mammary carcinogens can damage DNA. Those are a classical carcinogen like ionizing radiation or medical radiation. Then endocrine hormonal active chemicals or endocrine disruptors can act later in life when there's already a tumor as tumor promoters, and they can also act early in life like DES, or diethylstilbestrol, to affect development and alter susceptibility to a carcinogen and to other estrogen action. So those are our 3 sort of key mechanisms, and then we think about what are chemicals that we're exposed to that operate by those mechanisms. So these are some examples of that first mechanism. So mostly classical mammary carcinogens that cause tumors in an animal study. Ionizing radiation, some constituents of gasoline and auto exhaust, some pesticides, some flame retardants, some disinfection byproducts from drinking water, and so on. Now, I'm going to go back to that later because we just released a big paper about those kinds of chemicals. Also pointing out that the breast is very vulnerable, particularly vulnerable to those carcinogens as well as to hormonally active pollutants before birth and during puberty and pregnancy. The breast has a very long period of development. It's not actually fully developed until the end of the first full-term pregnancy, when the cells develop the ability to make milk and lactate. And so that long period before the first full-term pregnancy is a period during which the mammary cells are more vulnerable to classical carcinogens like X-rays. Three major authoritative kind of government reports have come out recently, all sort of supporting this line of thinking, and all sort of identifying the fact that we need chemicals testing to prevent breast cancer. We need to identify ways to predict chemicals of concern. The first is the President's Cancer Panel, which I mentioned before. The Institute of Medicine released a report, Breast Cancer and the Environment, in 2011, and the Interagency, U.S. Interagency Breast Cancer and Environment Coordinating Committee also did a report. So as I sort of mentioned before, there's growing momentum for this idea that we have to come up with better ways to predict and prevent. I'm often so I'm trying to tell the story really. What does chemical safety testing have to do with breast cancer? And so the things that we think are important for chemicals should be evaluated for safety before they're put in consumer products, for example, or widespread exposure. And we want to make sure the tests are relevant to breast cancer, that they're designed so that they'll pick up mechanisms that are important to breast cancer. And this is both what I was talking about in terms of the three mechanisms, but also Meg is going to, when she carries on in the second half of this, talk a lot about how we think about making sure the tests are relevant to breast cancer. We're using animal models and also in vitro or cell-based models to try to identify potential carcinogens and reveal mechanisms. And these are the same techniques that are used for drug discovery and as well as chemical safety testing. We have previously identified 216 chemicals that cause mammary tumors in rodents. So these aren't, for the most part, these don't include some hormonally active chemicals, but these include more traditional carcinogens as well as some strong hormones like hormone replacement therapy. And we recently published a study focusing describing how to measure those many of these chemicals in people with the hopes of encouraging more study in humans of these exposures, both so that we can reduce exposures now to these chemicals that might cause mammary tumors, that might cause breast cancer, based on the fact that they do cause mammary tumors in rodents. And the question, of course, is, well, rats aren't people, so are these really predictive? And so part of the paper, and this is, if you want to read more about it, I would go to this 2014 Environmental Health Perspectives paper. But we basically looked side by side at evidence in humans and evidence in animals for agents where there is both kinds of evidence. And so the pink and orange are showing where there's a positive association, and you can see generally between the rodents and the humans is consistently positive, or if it's blue, consistently negative. And only in for a couple pharmaceuticals that were tested did we have some inconsistency. So that's encouraging in terms of thinking about our tools and how good are they. This is more information about the paper that we just released, and where again we're looking at a systematic approach to thinking about chemicals that are priorities for breast cancer research and prevention. We're also hoping to minimize use of animals in testing and also minimize the expense of using animals in testing, because it's extremely expensive, and working on another project to develop more cell-based safety-based testing that can help identify chemicals that would be likely carcinogens. Sorry, it takes a, oh, there we go. Whoops. Because chemical exposures are so widespread, even a small increased risk or a small influence on cancer risk touches many lives. And so, so even if the incremental increase in risk from a particular exposure may be small, with so many people in the U.S. exposed for many environmental chemicals, it can, from a public health or population standpoint, have a significant public health impact. One of the outputs from our most recent paper is a list of these are some exposures to chemicals that are likely breast carcinogens or possible breast carcinogens. And people often want to know what they can do. So these are some idea of lessen exposure to fumes from gasoline and diesel exhaust or other fuel combustion, and also to support anti-idling and fuel efficiency regulations, because those would also reduce exposure. Sort of related and not on here is like don't store gasoline in an attached garage or basement, and to try to use electric or manual sort of lawn appliances rather than ones that use gasoline, to try to minimize their exposure to the fumes from gasoline and exhaust. can use a vent fan when you cook. Find a dry cleaner. It doesn't use perc or other solvents, so ask for wet cleaning, which is apparently better on your clothes too. To help keep your drinking water clean by protecting source waters, but also you can limit amount of either pollutants or drinking water disinfection byproducts by using a drinking water filter. To avoid stain-resistant chemicals for rugs and furniture and fabrics, and tell retailers and manufacturers that you don't want perfluorinated compounds or PFCs in your home or in the Arctic, which is where these all build up. Several flame retardants that are used in furniture foam are also contain some carcinogens and so on. But again, none of us can prevent breast cancer alone, so there's a lot of collaborative action that we need to do to help support these initiatives. On our website at Silent Spring, there are a lot of resources, our papers, and general information for the public, and more tips and more suggestions for how to take action. I'm having a little trouble progressing it to the next slide here. I don't know.
Great. Thank you, Ruth Ann. I'll progress it.
Okay, thank you.
There we go. And now I'm going to hand it over to Megan Schwarzman, who's going to talk a little bit and present her piece.
Thanks very much. It's a good combination because Ruth Ann sort of already set the stage for the project that I wanted to present to the gathered, if disparate group. So, Ruth Ann mentioned the idea of trying to recognize carcinogens now, not 60 years from now. And that's sort of all of what this project was about. And in order to do that, she's already acknowledged that we have to both increase chemical testing and make sure that the chemical testing that we do is relevant to breast cancer. And so faced with this suggestive evidence of the role of some chemicals in raising the risk of breast cancer, and also faced with really significant data gaps for all the reasons that Ruth Ann outlined, that we haven't traditionally studied women, and also how difficult it is to come up with human evidence and the general under-testing of chemicals. The California Breast Cancer Research Program, CBCRP, funded a project to investigate how best to test chemicals to understand their role in breast cancer. So I led this project along with Dr. Sarah Jansen, who's a physician and at the time was working as a senior scientist for the Natural Resources Defense Council. Let me make sure I can control the slide. That looked like it worked. And our core question was this: what body of information obtained using existing test methods could best identify chemicals that may increase the risk of cancer, of breast cancer specifically? And the emphasis was on the existing test methods for exactly this reason that Ruth Ann raised of we don't want to wait another 60 years until we have the perfect tests to identify breast cancer, but using, or identify chemicals that could cause breast cancer, but using the tools that we have available now, what's the body of information that would be most useful? So we designed the project to do three things, basically. And as soon as that slide changes, those three things will pop up. It doesn't seem to be advancing. There we go. One was to design an approach for identifying chemicals that may contribute to the development or progression of breast cancer. So we weren't testing chemicals. We were trying to pick the ways of testing chemicals that would work well to identify breast carcinogens. And the second was to identify research needs and recommend improvements of existing test methods. It's so tempting in this field where there's so little information about chemicals and the tests are often so imperfect to only focus on needs and the gaps. And that's why this was actually our second goal, specifically not our first. The first was really to working with what's available to us now, what can we do to identify chemicals that can contribute to breast cancer. And the third was thinking that basically this process could be a model, if it worked well, that could be applied to other disease endpoints with the ultimate aim of producing a comprehensive approach for identifying hazardous chemicals. So we might take the same approach and look at chemicals that might cause asthma or chemicals that might alter neurodevelopment, and we could start to see over time which of the tests are really common among a lot of disease processes and which are unique to, say, breast cancer or neurodevelopment, and if we didn't include them, we might be missing chemicals that contribute to that disease. So to do that, we convened an expert panel, and there was it was a very interdisciplinary panel and a whole range of experts, from people who are experts in breast cancer biology to toxicologists, epidemiologists, folks who have experience with risk assessment in a policy setting, in a regulatory setting, folks who understand chemicals policy and the chemicals policy changes at the federal level as well as the state level, and also community advocates. So we had a very diverse panel. And we designed the project around 5 specific steps that I'll lay out here, and then I'll go into each of them in more detail. The first was to identify toxicity endpoints, and that term really describes a change in a biological process that would increase the risk of breast cancer. So a biological process that's going along normally and then is altered, say, by a chemical, and is there an endpoint there that we could then detect with chemical testing. The second step would be to identify the toxicity testing methods that would detect chemicals that alter those biological processes that we've determined are relevant to breast cancer. The third step was then to arrange those tests into some kind of logical approach, and that was propose an approach for prioritizing chemicals to test and then for testing chemicals for their ability to raise the risk of breast cancer. And we called this approach, once we came up with it, the HIA-BC, which is for the Hazard Identification Approach for Breast Carcinogens. And I'm introducing that acronym here because it shows up in the last 2 steps. This was the main sort of product of our project, was this hazard identification approach for breast carcinogens. And we asked 2 questions of the HIA-BC once we'd come up with it. One was, does it work? So step 4 was to pilot test the proposed hazard identification approach for breast carcinogens by investigating how well-studied chemicals would perform if tested. So we didn't have the time or the funding to test chemicals, but we looked to the literature for well-studied chemicals and said, okay, if you put them through this battery of tests, how would that turn out? And then the second kind of inquiry that we made about the hazard identification approach was, how do the assays, the tests that we called for in the hazard identification approach, compare with those that are called for in the federal chemical testing initiatives? And I'll talk more about those chemical testing initiatives as sort of context for the playout of the project. But that's an overview of what we did in the project, and I'll show you the findings now, basically step at a time. So this first step was to figure out what events in biological processes are associated with the development of breast cancer. And this was a big brainstorming session that we did with the whole panel over a day and a half, trying to get down into a big Excel spreadsheet all of the things that can change that we have some evidence linking to breast cancer. And we organized them ultimately into sort of different levels at which they happen, cellular and molecular events, and those are obviously things that happen at the cell level, and they might be genotoxicity, like changes to DNA, or it might be something like a little bit less direct, like immune modulation, where you've altered the way the immune system works and it can't respond and correct damage that's done by a carcinogen, and that tends to lead to cancer. Various things like that that happen at cellular levels, and then sort of parallels at the tissue level, and then parallels at the at a much more general level of sort of susceptibility factors, and this is again less direct evidence, but things like obesity, where we know that obesity is linked to higher risk of breast cancer. And the premise for this was that in identifying chemicals likely to increase the risk of breast cancer, we should investigate those that are associated with general carcinogenic mechanisms. Also, so these actually map to the 3 categories that Rufan named, so sort of classic carcinogens, and also chemicals that increase estrogenic or other proliferative effects on breast tissue by any mechanism, and finally those that alter development of the mammary gland. So the second step was to then, we sort of lined up, once we'd come up with all of those events that can happen, that detectable events in biological systems that can affect breast cancer risk, we lined them up along the top of a large table, and this is just a mock-up of the table, and it has some example things in there, but just it was a sort of giant spreadsheet. And on the left-hand side, we described the sort of system in which that could be detected. So in silico is sort of a shorthand for saying computational toxicology, so comparisons that are made by computers based on chemical structure. In vitro would be cell-based tests, and in vivo is tests that are done in animals. And epidemiological evidence comes from studies of people. So we then populated this large table. The cells in the table then contained basically the tests, the assays, the ways that you could test to see if a chemical would have this effect. And once we had that large table of assays, tests, we were able to organize them into the hazard identification approach for breast carcinogens, the HIA-BC. And I'll show that to you in each of its 3 parts, but I wanted to give an overview first. It consists of 3 parts. The first is a prioritization step. And this is based on the premise that there's tens of thousands of chemicals in commerce, most of which have very little toxicity data available, and that it's sort of a non-starter to say we have to test all of them from the get-go. So is there a way to prioritize and test the ones that we're most worried about first? And then we divided, once you pass through the prioritization step, then the tests for the hazard identification approach are divided into 2 sections, those that can be done in cell-based tests, the rapid screening methods, and those that at this point still depend on in vivo studies, animal studies, to get the evidence. So the first step is the prioritization approach, and I won't go through all the details in each of these, but just to generally say that we divided it into 2 sections. One is how likely is it that people are to be exposed to this chemical, that's the exposure potential, including that this is a chemical that might end up particularly in fat tissue, which tends to be in breasts, among other places. And also, are there early hazard indicators? So are there structural similarities to other carcinogens? Is there something that has a metabolite that we know has some endocrine activity that would trigger an interest in further testing for its ability to affect breast cancer risk? So then the second level of the HIA-BC was the rapid screening, the cell-based tests that were meant to look, again, for these sort of 3 categories: genotoxicity, endocrine disruption, and the cell cycle changes has to do with some hallmarks of cancer that are the tendency of cells to become immortal, basically, when they are carcinogenic, when they've been mutated. So these are the examples of activity that you could test for in cells that might suggest that a chemical would contribute to breast cancer risk. And the final piece of the hazard identification approach is studies that still have to be done in animals that we haven't found a way to do in cells yet. And this includes more endpoints, the genotoxicity, the cell cycle changes again, endocrine disruption effects, but also some of the more developmental effects. That's where we have precursor changes, biomarkers, and induction of mammary gland tumors. So these are some of the things that you can really only detect, like altered breast development or altered mammary gland development. You can really only see if you look at the whole tissue at this point. So that was the HIA-BC as we came up with it, and a couple of notes on it just for clarity. One is that the panel was recommending endpoints to test rather than a battery or a list of specific assays, specific tests. And that just has to do with how quickly the field of toxicity testing is evolving. You know, every month there will be new methods for testing a particular endpoint, and that the endpoints remain steady. We just may get better at testing for it. So this was a way of having it not be instantly outdated. And the other was just a note that we're moving as a society, as a scientific society, away from, as much as possible, whole animal tests towards cell-based tests, and for a couple of reasons. One is to not use as many animals, and the other is that these high-throughput screens often can be done much more quickly. They can be automated, and they can therefore test thousands of chemicals and at many doses. So often we're limited to just a couple doses with animal tests. And there's also the potential to address some metabolic differences if you can test the metabolites of a chemical in a cell-based test. So the panel just wanted to acknowledge that there's a move in toxicity testing toward these mechanized high-throughput screens, and to support that move at the same time as acknowledging the HIA-BC doesn't have a lot of those in it because it's based on currently available tests. So then once we'd come up with the HIA-BC, the hazard identification approach, we wanted to pilot test it. We wanted to say, okay, is this valid? If we took some well-studied chemicals and put them through these tests, would they have the result that we would expect? And so what we did is, you can see the categories in bold. We found some chemicals that fell into each of these categories, chemicals known to cause breast cancer in women, those with some evidence of breast cancer in women, those where we don't have human evidence, but we have evidence of their carcinogenicity in animals, specifically mammary tumors, and chemicals that had suggestive evidence. These might be associated with breast cancer, but it's not definitive. Substances that we know are carcinogenic but aren't linked to breast cancer that we know of. And also chemicals that are fairly well studied and don't seem to be connected to any cancer. So, and then what we did is, with this list of chemicals, we did a literature review on each of them and completed a table that's too big and complex to show here, but I'll summarize for you basically a couple of kind of the key findings. One is that we confirmed that some chemicals thought to be breast carcinogens, such as vinyl chloride or benzene, mainly act through genotoxic mechanisms. So this is like damage to DNA, some of the classical carcinogenic effects. Others mainly act through hormonal mechanisms. So some examples of this are like dioxin, or the TCDD that is there, or the conjugated estrogens. And what this confirmed for us was that you can't overlook one of those categories of endpoints, and that just having some genotoxicity data on a chemical might not let you off the hook with breast cancer because endocrine-mediated changes are so important in breast cancer. Another significant finding of this pilot test was that for even this group of relatively well-studied chemicals, there were many data gaps. And that actually made it difficult to perform this validation step because not all the tests have been conducted for all the chemicals, and the information just wasn't at hand. So the extent of the data gaps actually made it impossible to do what you would want to do with the HIA-BC, which is to assign levels of certainty to tests in the HIA-BC. That is, you'd be able to say, well, if you get a negative on this and a positive on that, then you have this % chance of being a breast carcinogen. And in fact, we're not there yet. There's so little data on even these relatively well-studied chemicals that kind of algorithm, that kind of testing algorithm is still out of our reach. And the final step that I talked about sort of the questions that we asked based on the HIA-BC was to compare the tests that we called for in the HIA-BC, the endpoints we thought were important to study, with the endpoints that are included in the federal testing chemical screening initiatives. So to back up for one moment, the first two of these initiatives, ToxCast, that's run by the Environmental Protection Agency, and Tox21, that's an interagency screening program that involves the National Institutes for Environmental Health Sciences, the National Toxicology Program, and EPA. They're developing rapid, often mechanized robotic methods for screening large numbers of chemicals. And although they have a lot of promise, specifically the speed and volume and cost effectiveness, they're inherently limited to the assays that they use. So if right now most of the tests that these national programs use are sort of off-the-shelf, commercially available batteries of in vitro screens, and they may not be specific for the particular tissues or disease endpoints such as breast cancer. So there may be a panel of genotoxicity batteries which may be relevant to breast cancer and they may not entirely. And so that was, it was part of the rationale for this project in the first place, in that we could identify, at least at a conceptual level, important gaps in biological pathways that are not addressed in these federal chemical screening initiatives. If we started with the disease, as we did, and worked backwards through the known and suspected disease pathways, and ultimately to this list of endpoints that could be studied, and that we could compare that with the tests that we thought were important in the HIA-BC, we could compare those and sort of circle back and evaluate how well the endpoints we identified are represented in the national testing initiatives. The third piece on this slide is the endocrine disruptor screening program, which is developed to validate in vivo, so animal-based tests for endocrine disruption, and even some of those actually require modification to make them relevant to breast tissue. So we did this, a comparison actually that Ruthann headed up with some of her staff to look at the tests that are in the HIA-BC and compare them to the federal chemical screening initiative. And it led to some specific recommendations that we could make to the staff of the screening programs about endpoints that are not, at this point, currently well covered in those federal screening initiatives that should be if they want to be complete about looking for breast carcinogens. So in conclusion, the panel, after this whole exercise and a year or so, we made some final recommendations, and they're all sort of in this framework of that we need more chemical toxicity testing, and that it and the public policies that require it can inform breast cancer prevention efforts by identifying chemicals that may raise the risk of breast cancer, and that we should keep a few things in mind with that. The first is that it should include this range of endpoints that we've discussed before. The second is that we need to design and conduct toxicity tests to consider both the timing of exposure, so exposure that has different effects depending on the timing, including exposure during sort of developmental window, susceptible windows of development. And also that they need to consider underlying susceptibility factors. So there's a lot of genetic variability, and we differ in how well we can repair DNA damage or metabolize certain chemicals. We also vary as a population in what else we're exposed to, either because of where we live or what work we do, and that the toxicity testing needs to take that into account when we're designing it. And additionally, that of course we need additional research, and it should do a couple of things. It should increase our understanding of the biological pathways associated with breast cancer, because we found a lot of gaps in that area as well. We should adapt current testing methods to improve their relevance for breast cancer, so there may be some screening methods that are established and can be performed by labs but aren't that relevant for breast cancer, and that we also need new toxicity tests for the endpoints that we don't have good ways of studying. And that includes identifying early events is something that the panel really wanted to call out, such as altered mammary gland development, things that occur really well upstream of tumor formation, and that if we understand those pathways better, that enables the scientific community to develop assays that would test if chemicals contribute to those upstream events. And this actually led to, this project informed a second request for proposals from the CBCRP, the California Breast Cancer Research Program, to develop some tests for the endpoints that we had identified in this project as needing specific assays. And finally, that going back to our original goals, that it would be possible to apply a similar process to other disease endpoints, and that that kind of process could inform the development of a comprehensive approach to chemicals of concern, and that could be incorporated into data requirements in new chemicals policy. So finally, we have a fair amount of output from the project. I can't seem to advance it to the last slide. There it is. Whoops, I went too far. There it is. So the full report from the panel's meetings is available online at that first link. Perhaps of most interest to this group are the advocacy materials that we put together. So there's a 10-page booklet that's sort of a summary of this report, of the full report, and it's also translated in Spanish, and a 2-page fact sheet that's a shorter version yet, translated into Spanish, Chinese, and Korean. And they're aimed for the advocacy community to be able to help you go out and talk to people about increasing chemical testing, putting testing requirements into chemicals policy. The panel felt very strongly about the need for chemicals policy reform because of the significance of the data gaps and how much that impairs our ability to understand now what chemicals might be contributing to breast cancer. So with that, I'll turn it back to Saru.
Great. Thank you, Megan, and thank you, Ruth Ann, both for your wonderful presentations. And in a minute, we're going to open it up to questions. I wanted to just take a minute to talk about sort of where we're at right now in terms of chemical policy and chemical safety reform. Now, in the last 2 years, there have been 3 bills introduced, and those are the Safe Chemicals Act of 2013, the Chemical Safety Improvement Act, and the Chemicals and Commerce Act. And actually, only one of these, the Safe Chemicals Act, is actually a strong bill that would provide the reform that we need to protect public health. But what we're seeing is that there's a real growing momentum to update the Toxic Substances Control Act from 1976, also called TSCA, with the EPA, with environmental and public health organizations, all who really care about public health. Unfortunately, this growing momentum has also been happening with the chemical industry, and corporate lobbyists have also decided they want to update the TSCA, Toxic Substances Control Act, but their motivation is to weaken the current reform that we have, or the current legislation that we have, and provide more protection for the chemical industry. So what we need is our representatives really need to hear from us, and Breast Cancer Action will continue to work with our partners to make sure and prevent any bill that would weaken our current chemical safety bill that we have, and we will demand that any reform bill really puts public health before chemical industry profits. And you can go on our website and take action to also tell your senators that you feel the same way. So there were a number of resources mentioned in both Ruth Ann and Megan's presentations, and I wanted to just talk over them a little bit, and I will also be sending these in a follow-up email if you're not able to write all of these websites down. So for real detailed science reviews, you can check out an online database at the Silent Spring Institute's website, which is on the screen. There's also, as Megan mentioned, the Center for Occupational and Environmental Health at UC Berkeley has a really great chemicals and breast cancer advocacy materials, and you can find them there. And then a few other resources that were mentioned, if you're interested in sort of reading more, is the Institute of Medicine's report on breast cancer and the environment called a Life Course Approach. There's the President's Cancer Panel report in reducing environmental cancer risk, what we can do now. And last, the Interagency Breast Cancer and Environmental Research Coordinating Committee's report, Prioritizing Prevention. And again, I'll be sending these out in a follow-up email. So we're going to open it up in just a minute. I just wanted to talk a little bit more about how you can get involved with Breast Cancer Action. If you're interested, you can sign up and become a member. You'll get news and action alert emails from us to keep up to date on all the issues related to breast cancer. You can join us on Facebook and Twitter to connect with others and help change the conversation about the breast cancer epidemic. You can also help others get involved. You can tell your friends, your coworkers, your family about this webinar and ways that they can get involved. And last, you can donate to support our education and advocacy work. So again, I want to give a really big thank you to Megan Schwarzman and Ruth Ann Rudel for their wonderful presentations today. And now I want to open it up to your questions. And people have been typing in questions as we've been going along. So I'm going to go through and pose questions. So there was a question that someone asked. They said, why do regulators and politicians insist on statistically significant results from breast cancer epidemiological studies before taking action when the weight of scientific evidence or health risk assessments are used in other regulatory contexts? And I'll put that out to either of you or to both of you.
This is Ruth Ann. Well, I think that in some cases there is regulatory action before there's human evidence, and then in other cases there isn't regulatory action even when there is human evidence, and then we have everything in between. I think a lot of times it really ultimately depends on the, it ends up depending on the economic interests and the costs, the apparent costs on the of the regulatory change, and the and who's speaking the loudest. I think that, for example, what's motivating some action towards chemicals, the chemicals policy reform, which Saru was talking about, is that in the absence of any federal regulation, two things have been happening. States have started enacting their own bans on specific chemicals and restricting uses of chemicals, and that's creating a patchwork effect, which chemical manufacturers don't like at all, and so they've started to become interested in a chemicals policy reform that would even things out again, so that they wouldn't have different standards and requirements in different states. And the second thing that's been happening is that people have been voting with their pocketbook, essentially, by putting pressure in the marketplace, asking manufacturers for certain kinds of chemicals that don't contain, certain kinds of products that don't contain, chemicals that are particularly concerning, and in response to that, the manufacturers of the products have turned around and said, ask the chemical manufacturers for, have started trying to reformulate, basically, to limit use of the most problematic chemicals. So, and it's those kinds of pressures actually are, I think, maybe a lot of what's fueling the any industry interest in chemicals reform. And the other part of that question maybe is that sometimes in the clinical realm, doctors can be less familiar with how we use animal evidence and other predictive evidence to make guesses about which chemicals might be carcinogens, and they might be more used to looking at randomized trials, like for drugs in humans, and so since that's their framework and they don't see that kind of data for environmental chemicals, there can be some sort of residual skepticism in the medical community. I think mostly really just due to a lack of familiarity with the predictive methods that have to be used because we can't do randomized trials with chemicals.
Right. Thank you, Ruth Ann. There was a question by someone who wanted to know in your studies, and I'm not sure if this is directed to Megan or Ruth Ann, so whoever wants to answer it, please feel free. But they wanted to know if you found a link between certain types of chemical exposures and different types of breast cancer, if you were looking at sort of any sort of specificity with the different types of breast cancer related to different kinds of chemicals or chemical exposures.
This is Meg. I think Ruth Ann can probably say something about this with regard to her recent paper, but I'll just clarify from the Breast Cancer and Chemicals Policy Project. I would say, we were really trying to cast the net broadly so that we were making sure we were calling for chemical tests that would address all of the ways that breast cancer can be caused, because the panel was very clear and wanted to acknowledge that breast cancer is not actually one disease, and that it's many different diseases. It's at least 4 or 6 different diseases, and that there are probably different mechanisms at play in different types of breast cancer. And so although it wasn't within the sort of purview of our project to start assigning which kinds of chemicals might be most relevant to which kinds of breast cancer, the panel did very much want to call out the notion that breast cancer is actually a range of diseases, and it's one of the reasons that that broad range of mechanisms needs to be tested for when chemicals are evaluated.
Great. Thank you, Megan. Ruth Ann, did you want to say anything on that?
Oh, that was a great answer.
Okay. So we're running a bit over time, but I want to take a couple more questions, and then we'll wrap it up in a couple of minutes. There's a question, I think this is directed to you, Megan. It says, do you include developmental epigenetic changes in the genotoxicity category? And if that's not where they are, where would those show up?
That's a good question. I actually had taken them off of one of the slides because it was too much to explain what developmental epigenetic changes are, but the panel certainly did include them in the HIA-BC, or they felt like they were an important endpoint to evaluate. The problem is that there aren't a lot of assays for those changes now, and we don't know what to do with the results yet much. And so there was a lot of discussion in the panel about the role of epigenetic changes in breast cancer. And in the longer version of all this that's spelled out in the reports, we discuss epigenetic changes and the importance of developing assays for them and investigating more about their role in breast cancer. So where they really fall right now is in the calling for more research on the pathways, the biological pathways that contribute to the development of breast cancer, because we suspect that developmental epigenetic changes play a role, and probably a lot of them are they're hormonally mediated, but there may be other mechanisms, and that so really where they fall into is in the specific calls for additional research.
Great, thank you. And then there was, I think, maybe a clarifying question someone was asking when you were referring to ionizing radiation. I think this was directed to you, Ruth Ann. They wanted to know if this included low-dose ionizing radiation, such as mammograms, or if it was referring to sort of high-dose ionizing radiation.
Ionizing radiation is well, there may be some disagreement with whether low doses have effects, but I'd say the general consensus is that, it's what you call it's a stochastic model, like a probability-based model. A little bit of exposure would be like buying, one lottery ticket, and a lot of exposure would be like buying 100 lottery tickets. Either way, you have a chance of winning, but your chance is higher the more exposure you have. And for breast cancer in particular, the chance of winning is also strongly influenced by your stage of development when you have the exposure. So early in life, the breast is much more vulnerable to something like ionizing radiation than later in life when the tissue is more fully differentiated and developed.
Great. Thank you. So the last question, there are a number of questions about getting these slides after this presentation. And so I want to tell everyone that we have recorded this webinar, and we'll put a recording on our website along with a PDF of the slides. And those are usually posted about a week after the webinar airs. So I'm going to close it with that. I want to respect everyone's time and thank you all for your thoughtful questions and comments. And if we didn't have a chance to answer your question today, you can follow up with us at info@bcaction.org. And I also want to let you know we will be sending an email with a short survey where you can provide some feedback on this webinar. Thank you all so much for attending, and have a wonderful rest of your day.